Hello students, welcome to the biology class. How are you all guys? Hello everyone, Pratima, Purva, Mansi, Gopal and Gaurav. Thank you Gaurav for the sanitizer. Thank you again. Hello Nilima. Okay. अच्छा प्रतिमा नो दादागिरी बेटा राइट पहले तुमसे सब डरते हैं thank you very much Gaurav. All right, so today I'm taking class from Pandit Colony. All right, yes, I'm standing here. You must be noticing the changes, right? The area here is quite short, and there in the college it is wide, quite wide, right? Definitely आप सबको डराते हो तो डरेंगे ना बेटा. Hello Rohit, hello Neha. Okay, so let's quickly start our today's topic. In the last class, we have just discussed about the different type of pollination, right? In short, self-pollination, cross-pollination, right? And then we have discussed about the various pollinating agencies also. Pollinating agencies mainly are of two types. Number one, biotic and then abiotic. Hello, Jamila. Okay, so now today we need to discuss in detail of all the type of plants and their characteristic features. If they are pollinated with wind, then what special characteristic feature they are having? Okay, so we need to discuss all this. So let's quickly start. First, we will discuss the characteristics of plants which are pollinated through wind. Okay, and what do we call the plants which are pollinated with the wind? Yes, yesterday we have discussed. Quickly write in the chat box, what do we call the plants which are pollinated with the help of wind? Yes, what do we call those plants? Yes, that is anemophily, pollination with the help of wind, and they are anemophilous plant. Okay, so first we will discuss the characteristic features of anemophilous or anemophily, right? अच्छा, अब जब पॉलिनेशन विंड की हेल्प से हो रहा है, मतलब जो ट्रांसफर है पोलन का टू स्टिग्मा विंड के हेल्प से, बहुत अमाउंट में पोलन बनाने की जरूरत होगी ताकि जैसे ही विंड चलेगी तो उसके साथ साथ बहुत सारे पोलन जो है वो मूव करना स्टार्ट करेंगे. अगर कम होंगे तो चांसेस आर लेस कि पोलन जो है वो स्टिग्मा तक पहुंच पाए. इसीलिए नंबर वन कैरेक्टरिस्टिक फीचर इज दैट pollens are produced in large number. Pollen are produced in large number. Okay, yes. Next characteristic feature, yes, you have written correctly. These are the examples of anemophilous plant. Next feature is that their flowers are very small actually, but number of flowers are many, so their flowers are more small but more in number. Okay, that is second characteristic feature. जितने ज्यादा फ्लावर होंगे डेफिनेटली उसमें एंथर होंगे एंड दोस एंथर विल प्रोड्यूस मोर नंबर ऑफ पोलेंस, राइट? एग्जैक्टली. नेक्स्ट कैरेक्टरिस्टिक फीचर इज दैट whatever the non-essential part present in flower that are not present, जितने नॉन इसेंशियल पार्ट प्रेजेंट होंगे उतना रेजिस्टेंस प्रोवाइड करेंगे वो पोलन के स्टिग्मा तक पहुंचने के लिए, राइट? What happened? प्रतिमा, what you are writing बेटा? गौरव लिख रही हूं मैं अभी. Many more. Yes, there are many more. Okay, not an issue. Pay attention. So less or no presence of non-required part of the non-essential part of the flower. Okay. Coming to the next feature. अच्छा, यहाँ पे एयर विंड की हेल्प से पॉलिनेशन हो रहा है, तो इसके फ्लावर्स को ज्यादा ब्राइट कलर्स के पेटल्स भी नहीं चाहिए टू अट्रैक्ट दी बायोटिक एजेंसीज, इंसेक्ट्स, बर्ड्स, ये सब नहीं होगा, तो इसके फ्लावर कलरलेस होंगे डेफिनेटली. फिर नेक्टर, स्मेल, ये भी नहीं चाहिए क्योंकि विंड को इन सब से कोई लेना देना नहीं है, ए बायोटिक फैक्टर. सो दैट्स व्हाई देयर फ्लावर्स आर small, colorless, smellless, do not have any nectar, right? Okay, चलो ठीक है, झगड़ा बाद में, अभी पढ़ाई करते हैं, राइट? आई गेस ये टॉपिक आज थोड़ा जल्दी कंप्लीट हो जाएगा, तो वी हैव प्लेंटी ऑफ टाइम फॉर झगड़ा, राइट? ओके. Next, pollen grains are very light, right? They are very lightweight, so that they can easily move with the help of wind. बहुत lightweight होंगे, अगर heavy हुए तो wind की help से move नहीं कर पाएंगे. Plus, some pollens are winged. उनके पास ऐसे structure होते हैं wing-like, which helps them in moving with wind, right? So they are light-weighted, and some of them are possessed with wings. Okay, yes. And the example of winged pollen is found in Pinus. All of you have studied about Pinus, the gymnosperm. It is having winged pollen grains. Okay, all right. Now, अब हवा की मदद से ये इधर उधर ट्रांसफर हो रहे हैं, तो डेफिनेटली अगर ये किसी लेट्स से ह्यूमिड जगह पर जाकर थोड़ी देर रेस्ट कर रहे हैं और ह्यूमिडिटी ने इनको खराब कर दिया, वाटर ने इनको खराब कर दिया, तो फिर पॉलिनेशन नहीं हो पाएगा. दैट्स व्हाई दे शुड बी अनवेटेबल, मतलब ये इनके ऊपर पानी ठहरना नहीं चाहिए, राइट? दिस इज अनदर कैरेक्टरिस्टिक फीचर ऑफ plants or pollens which are transferred with the help of wind. Okay, yes, so they are unwettable and non-sticky. Non-sticky होंगे तभी अच्छे से मूव कर पाएंगे, स्टिकी होंगे तो किसी भी स्ट्रक्चर से जाके स्टिक हो जाएंगे. All right, and pollination cannot be done then. All right. Okay, so these are some कैरेक्टरिस्टिक फीचर्स. अच्छा, एक और फीचर ये है प्लांट का कि चलो पोलन आपने बहुत अमाउंट में बना दिए, सब कुछ कर दिया, लाइट वेटेड कर दिए, नॉन स्टिकी कर दिए, विंग्ड कर दिए, बट इनको रिसीव करने वाला स्टिग्मा वो भी तो थोड़ा अडेप्टिव होना चाहिए, राइट? विंड के थ्रू पॉलिनेशन के लिए. तो यहां पे स्टिग्मा जो है वो ब्रांच्ड होते हैं, राइट? थोड़े से बड़े होते हैं, just to receive the pollen with the help of wind. Okay, so these are some characteristic features. Absolutely right, Jamila, you have written correctly. Now, अब क्योंकि ये तो थे इनके कैरेक्टरिस्टिक फीचर्स, अब इससे रिलेटेड कुछ अबनॉर्मलिटीज और सम डिजीजेस वो डिस्कस कर लेते हैं. अब ये हवा में उड़ रहे हैं सारे पोलन, जब भी इनका सीजन आता है, जनरली स्प्रिंग सीजन में ये बहुत ज्यादा होते हैं हवा के अंदर, राइट? तो सम पोलन कॉजेस एलर्जी इन सम पीपल. ओके, क्यों? क्योंकि इसके ऊपर एक एलर्जन रहता है, टफ मटेरियल रहता है, which I have already told you, sporopollenin, right? Other component can also cause allergy in people, right? तो ब्रोंकाइटिस की प्रॉब्लम देखी गई है जब भी पोलन जो है एयर में ज्यादा होते हैं, राइट? देन हे फीवर करके एक और बीमारी है रिलेटेड विद रेस्पिरेटरी सिस्टम, हे फीवर. यू मस्ट हैव स्टडीड अबाउट हे फीवर. अपन diseases chapter में पढ़ेंगे फिर से. All right. So these type of abnormalities, diseases can also be caused by the pollens. Okay. All right. Now, next we will discuss about hydrophily. Okay, plants which are pollinated with the help of water. Hello Prathu, you have joined very late, beta. Second, hydrophily, pollination with the help of water. Okay. Yes, the agency of pollination will be water. Again, we are discussing about abiotic pollinators, right? Okay. Now, in hydrophily, there are two different types of pollinations are found. Okay. One is simply the hydrophily, which is taking place inside the water. All right, inside the water. And second is epihydrophily. Epihydrophily. Now, epihydrophily is on the surface of water. All right. Yes, it is on the surface of water. So that is the difference. Whenever we are talking about hydrophily, in general we call it pollination with the help of water. But again, it is divided into two types: hydrophily inside the water and epihydrophily on the surface of water. Right. So in total, we will discuss the common characteristics of these plants. Number one characteristic: the pollen produced are not to be what we can say decayed. ये पानी में है तो जल्दी डीके होने के ज्यादा चांसेस है, बट इनके पास ऐसी प्रोटेक्शन होनी चाहिए कि ये डीके ना हो पाए. अब ये प्रोटेक्शन क्या होगी? There is a coating of mucus around the pollen, all right, which protect them from decaying because they are present in water. So number one characteristic is this. Number two is that if they are opting for surface pollination, then they should be floating. All right. अगर heavy होंगे तो they will drown, ये डूब जाएंगे और surface के ऊपर इनका pollination नहीं हो पाएगा. So they should swim. All right. They should be lighter so that they can swim. Right. Yes, they can also be unwettable, मैंने बताया, otherwise ये decay हो जाएंगे. Okay, yes. Now coming to the example is that hydrophily का example हो गया आपका lotus, water lily, water hyacinth, and epihydrophily का example हो गया आपका Wellisneria. Okay. वेलिस्नेरिया के जो स्टिग्मा होते हैं वो सरफेस के ऊपर होते हैं. लाइक दिस इज द वाटर लेवल एंड दिस इज वेलिस्नेरिया फीमेल प्लांट. दिस इज द स्टिग्मा. दिस इज मेल प्लांट, राइट. दिस इज मेल pollen producing structure. This will produce pollen which will float and then finally reaches to the stigma. Okay. Yes. So this is the example of epihydrophily. Example Wellisneria. Okay. Let me write the name also. Wellisneria. Right. Okay. Yes. Now many times what I was going to tell you. It is seen that nectars smell and different colors are also not found in the flowers which are pollinated with the help of water, because abiotic agency है, again इसको अट्रैक्ट करने की कोई जरूरत as such है नहीं. Okay, hello Harsha, joining very late beta. Not an issue, we are discussing about the characteristic features. Okay, so that was there. Moving on to the next कैटेगरी. अच्छा, नेक्स्ट में मैंने आपको बताया था बायोटिक फैक्टर्स. ऑलराइट, नॉट एन इश्यू. नाउ वी आर डिस्कसिंग बायोटिक फैक्टर्स. बायोटिक फैक्टर्स में मैंने आपको इंसेक्ट्स के बारे में बताया था. इंसेक्ट्स के बाद वी हैव डिस्कस्ड अबाउट बर्ड्स, बैट एंड स्नेल, राइट. तो दीज ऑल आर एनिमल्स एक्चुअली. ये सबके सब एनिमल्स हैं. दे बिलोंग टू एनिमल किंगडम. दैट्स व्हाई कलेक्टिवली pollination with the help of these agencies, with the help of these animals, is known as zoophily, right? Collectively we are placing it into zoophily. Zoo is related with animals, zoans, right? Okay, so next is zoophily, in which we will study about insects, birds and bats. All right, zoophily, pollination with the help of animals, right? Okay, guys, my voice is clear. Am I audible to everyone? Hopefully. Yes. Okay, that's great, Purva. So in zoophily, different types of animals are involved. Even lemurs, the primate, they are also involved in the pollination process, right? Other than that, some snakes, some lizards, the garden lizard, they are also responsible for the pollination process. Okay? Yes. So in that, first we will discuss about the entomophily. Entomophily. What is entomophily? Pollination with the help of insects, honeybees, wasps, etc. Right? Now, what are the characteristic features of flowers or plants which are pollinated with the help of insects? So, definitely they should have attractive colors. Petals should be very attractive in color, right? Yes. Red color के होने चाहिए, then blue color के होने चाहिए, so that insects can be attracted. Next is they should have pleasant smell, right? Pleasant smell to attract different insects, and they should produce nectar. Now, what is nectar? Nectar is a sugary solution, sugary fluid produced by flowers. All right, definitely it is used to attract the pollinating agencies. All right, birds, insects, etc. Okay, so what I was telling you, yes, they are brightly colored, they must produce nectar. All right, and they have a landing platform for insect. Landing platform, definitely insect will fly and then land on the flower. So they should have a landing platform. Either their petal size should be more, अगर छोटा सा flower है और attractive है, it not be ये insects के through pollinate नहीं हो पाएगा, right? There should be a landing platform also. Then what are the other? I have told you they should produce nectar. They should have a pleasant odor. Yes. And that's it. Okay. अच्छा, किसी किसी केस में एंटेमोफिलस प्लांट में सेफ एनवायरमेंट होना चाहिए एग लेइंग के लिए, राइट? मेनी इंसेक्ट लेज देयर एग इनसाइड द फ्लावर, ओके? While they are coming for laying egg, accidentally they perform pollination, right? So there should be a safe environment for egg laying in the flower, okay? So that insect can come and perform pollination. All right. Now, example of this last I have told you, egg laying environment is yucca and amorphophallus. All right. Yucca and amorphophallus. These are the two plants which are providing favorable environment for egg laying to the insect, right? Yes. All right. So that is there. Now, in entomophilous plant, there are different modifications. Okay. One of the important modifications we are going to discuss is liver mechanism. Okay. They have liver mechanism, also known as turnpike method, turnpike mechanism. ठीक है? अब होता क्या है इस mechanism में? First of all, the flower looks like this. This is the petal. Okay, and this is the calyx and pedicel. All right. Anthers are present like this, just like they are arranged like a liver. There is a sterile stigma, and this is the fertile stigma. This is fertile stigma, and this is anther. This is sterile stigma. Okay, so this is the arrangement actually. Now what happened? Insect come to suck the nectar or the fluid present in flower. Generally it moves towards this stigma, sterile stigma. तो जैसे ही वो insect, let's say यहाँ पे insect आया, all right, जैसे ही इसने इसके ऊपर प्रेशर लगाया, ये जो एंथर था ये टर्न होके और इंसेक्ट की बॉडी पे टच हो जाता है. All right, and all the pollens are stick to the body of insect. Okay, now this will move to another flower, and whenever it will reach to another flower, it will transfer these pollen to the fertile stigma. Okay, yes, it will transfer this to the fertile stigma. This mechanism is known as liver mechanism. All right, because anther and sometimes stigma are also like this. They have grown outside the flower and move like this whenever any insect comes, so that they can touch the body, and indirectly all the pollens present on the body of insect will be transferred to the stigma. This is turnpike method or liver mechanism of pollination, right? This is found in the plant salvia. It is found in the plant salvia. So you need to remember the example. Salvia के अंदर ये वाला mechanism मिलता है. Okay? Yes. Now, So that was about entomophily. Moving on to the next, that is ornithophily. We are in zoophily only. Next is ornithophily. Okay, so we are having Jayendra. जयेंद्र, इलेक्ट्रिसिटी आ गई लगता है आपके एरिया में. दैट्स गुड. नेक्स्ट इज ऑर्निथोफिली. ओके. नाउ ऑर्निथोफिली में सबसे इंपॉर्टेंट कैरेक्टर है पेटल्स का कलर, सो दैट बर्ड्स कैन बी अट्रैक्टेड. ओके. एंड उससे भी इंपॉर्टेंट कैरेक्टर है प्रोडक्शन ऑफ नेक्टर, द शुगरी सोल्यूशन. इन्हीं नेक्टर को कलेक्ट करने के लिए बर्ड्स फ्लावर्स के पास आती है एंड पॉलिनेशन परफॉर्म कर देती है. राइट? यस, इट इज विद द हेल्प ऑफ birds. Now, what are the characteristics? Definitely, the shape of flower is like tube. This is the calyx. Okay, and like this. Now, the birds which helps in ornithophily, they have a long beak just to collect the nectar released by flowers. Okay, long beak. Definitely this bird will be hummingbird or sunbird. Right? Yes, it should have tubular corolla. I have told you tube-like structure. एंड डेफिनेटली एक इंपॉर्टेंट और फैक्ट बताता हूं आपको. हमिंग बर्ड बहुत छोटी बर्ड होती है, राइट? बट एक बार में ये अपनी बॉडी के हाफ वेट जितना नेक्टर सक कर सकती है. ओके, लेट्स से अगर ये 1 kg की है तो 500 ग्राम्स ये वन टाइम में नेक्टर जो है कंज्यूम कर सकती है. एब्सोल्युटली राइट. ओके. Crow as such does not help in the pollination, though it's a carnivorous, we can say omnivorous bird, right? All right. So that is their example of common bird pollinated plants are coral tree, okay, bottle brush, agave, and bignonia. Bignonia is another important example which is pollinated with the help of birds, right? Yes, bombyx is also there. Okay, so that was about the bird pollinated flowers. Okay, now next type is chiropterophily. Which means pollination with the help of bat. Okay, absolutely. मैंने बताया आपको crow is omnivorous, basically carnivorous. उसका जो beak का shape है, it is made for tearing the flesh, right? मीट को टीयर करने के लिए बना है. That's why. Now, chiropterophily. अच्छा, chiropterophily का इंपॉर्टेंट कैरेक्टरिस्टिक फीचर ये है कि बहुत दूर दूर तक पोलन ट्रांसफर हो सकते हैं, कई किलोमीटर्स तक, राइट? इवन 1000 किलोमीटर्स दूर तक bats can transfer or transmit pollens, right? Yes, तो सबसे इंपॉर्टेंट तो ये हो गया. प्लस दूसरा कैरेक्टरिस्टिक फीचर ये है कि the flowers should bloom in night. or they should remain open during night, because bats are active during night only. They are nocturnal. So they should have and they should produce some kind of scent, either pleasant or unpleasant. Both will attract the bats. If unpleasant, yes, they should have a strong scent of any type, pleasant, unpleasant. Unpleasant will resemble like fermentation, and pleasant, there are different pleasant scent. So, these are some characteristic feature of plants which are pollinated with the help of bat. Okay? Yes. And next is Malacophily, that is with the help of snail. Definitely they should be present near water, because snail is also present near water. Okay? Yes. So these are some characteristic features we have discussed. All right. I hope they are clear to everyone. They are very simple actually. Now, moving on to the next topic. आपके बोर्ड से मुझे कुश्ती लड़नी पड़ रही है उसको क्लीन करने के लिए. Okay. Now, next topic is are some terms, important terms related with pollination. Okay, and after pollination. So, number one term we are going to discuss is declining. Okay, declining. In this, the plants are unisexual, so that self pollination is not at all possible, क्योंकि plants खुद ही unisexual है. All right, either they produce ovule or pollen. That's why there is no self pollination. No self pollination, as plants are unisexual. Unisexual. All right. Absolutely right. Now, the example of declining is in mulberry and maize, right? Mulberry and maize. Yes, I need to do dangal. I'm writing here mulberry and maize are the example of declining, because they produce unisexual flowers. All right, either they will have, yes, Jayant, we can say Vallisneria also. That is the case of aquatic plant. Okay. Now, next term is dichogamy. It is the condition in which flowers are bisexual, but maturity time is different. Hello, Varun. Yes, you are very late. You are very late actually. Okay, not an issue. Watch the recording. All right, I was telling you about dichogamy. In this, self-pollination is avoided because the maturation time of anther and stigma is different. Okay? Yes, male and female matures at different time. Absolutely right. And that is how self-pollination is avoided. Okay? Now, if anthers are maturing earlier than stigma, then it is known as protandry. Okay? If anthers are maturing early, then stigma, then it is known as protendry. मैंने ये साइन बना दिया यहाँ पे. वैसे भी pro means पहले and endry is related with male part, right? Yes. So that is protendry, and the example of protendry is found in sunflower and salvia. Salvia we have just discussed the turnpike method, liver mechanism. Coming to the second, when stigma matures before the anther, that is protogyny. Okay, female part are maturing earlier. Okay, and the example of protogyny is found in your plantago and 4 o'clock plant. जेनेटिक्स में आपने इसके बारे में पढ़ा था, रेड, व्हाइट एंड पिंक फ्लावर. Yes, that is Mirabilis jalapa. Okay, the 4 o'clock plant. Yes. Okay, so that was the second term, that is dichogamy. Coming to the third term, that is prepotency. All right, the third term, I'm writing it here. That is prepotency. All right. Prepotency is the phenomena in which anthers or the pollen of other flower are capable of germinating on stigma of another flower. Okay. This phenomena is prepotency. मतलब किसी दूसरे फ्लावर के पोलन, ऑलराइट, जाकर किसी दूसरे फ्लावर के स्टिग्मा पे जनरेट हो सकते हैं. ऑलराइट, ये जल्दी मैच्योर होके पोलन ट्यूब बनाते हैं. अगर सेम फ्लावर का होगा तो वो लेट जर्मिनेट होगा. इससे पहले दूसरे फ्लावर का जो पोलन है वो जर्मिनेट होके पोलन ट्यूब बना देगा. दिस फिनोमेना इज प्रीपोटेंसी एंड इट इज फाउंड इन एप्पल प्लांट. राइट? यस. All right, so that is prepotency. Next is self sterility. Okay, name itself suggesting ये होगा क्या? Self sterility. It means कि अगर same flower के pollen है तो they are not capable of germinating the pollen tube on same stigma, right? It is due to genetic incompatibility. Some genes are responsible for the germination of pollen tube. Those genes are non-functional in that particular environment. That's why self-sterility is there. तभी self-sterility वहां पर मिलती है. All right? Yes. And gene के नाम भी बहुत easy है. S1, S3 in pistil and S1, S3 in pollen grain. So you just remember S1 and S3 gene in pollen and S1, S3 in the female gamete. All right, the pistil. All right. And example of this self sterility or self incompatibility are tobacco plant, potato plant. All right, potatoes and tobacco. These two are the example. Absolutely right, Prathu. All right. Next term is heterostyly. Okay. Fifth is heterostyly. Okay. What happened in heterostyly? Hetero means different. And style is related with that part of pistil, the style, right? There are different type, different length of style in different flowers. Okay, so the length of style varies in different flowers. Okay, and that lead to the phenomena heterostyly. Okay, yes, in some style is shorter than anthers, in others style is longer than anthers, like this. So there is a variation in the length of style known as heterostyly. Okay, now moving on to the, okay, let me tell you the example of this. Heterostyly is found in the plant tristyly. Name of the plant is itself tristyly. Three different type of styles are found. Primula, the primrose, okay, and lythrum. Lythrum is another example. Yes, Jamila, absolutely right. Okay. Jayendra, why you have sent this teacups? चाय क्यों भेज रहे हो भाई हमारे लिए? Okay, not an issue. हम पी लेंगे चाय भी. Now, next term is hercogamy. Next term is the sixth term, hercogamy. Now, इसके अंदर डिफरेंट मैकेनिज्म प्रेजेंट होते हैं. अच्छा, इसका बेसिक मीनिंग क्या होगा? ऐसा मैकेनिज्म, मैकेनिकल ऑब्स्ट्रक्शन प्रेजेंट रहना फ्लावर में जिससे कि सेल्फ पॉलिनेशन नहीं हो पाए. ओके, यस. एग्जाम्पल ऑफ सेल्फ स्टेरिलिटी. Oh, so tea plant. Oh my God. Very good. All right. So there are some mechanical hurdles, mechanical procedures are present which prevent the process of self pollination. For example, पोलन जो है, जब एंथर मेच्योर होता है तो पोलन सीधे फ्लावर के बाहर शूट आउट कर दिए जाते हैं. राइट, सीधा एक जेट के फॉर्म में फ्लावर के बाहर रिलीज कर दिए जाते हैं. फ्लावर के अंदर कोई पोलन रहता ही नहीं है. राइट, सेम फ्लावर का. दिस प्रिवेंट द सेल्फ पॉलिनेशन. ऑलराइट, यस. एग्जाम्पल ऑफ दिस इज योर कैलमिया, ऑलराइट, साइप्रेपेडियम, calotropis and orchids. These all are the example of plants which are showing hercogamy, which prevents self-fertilization by different mechanical procedures. All right? Yes. Now, अब ये इतनी terms की ये सब कुछ self pollination prevent ही कर रही थी. क्यों self pollination prevent करना है? ऐसी क्या जरूरत है कि हमें सेल्फ पॉलिनेशन नहीं करवाना है, क्रॉस पॉलिनेशन ही करवाना है? कोई बता सकता है ऐसा क्यों कर रहे हैं हम? लेट मी टेल यू. यस, सेल्फ पॉलिनेशन will produce pure line, and pure line does not have any variation, whereas cross pollination will produce variation, and variation will leads to the increased immunity. All right, increased immunity towards the diseases. जबकि pure line जो है, they are susceptible to a particular type of disease, more susceptible to a particular type of disease, because धीरे धीरे उनकी gradually immunity low हो जाती है. Right? Yes. So that's why cross fertilization और cross pollination जो है उसको यहाँ पे बढ़ाया जा रहा है और self pollination को prevent किया जा रहा है due to these mechanism. Right? Yes. मतलब देखो, plants के पास brain नहीं है, फिर भी उनको पता है कि cross पॉलिनेशन ही ज्यादा इंपॉर्टेंट है, राइट? तो सेल्फ पॉलिनेशन को जो है डिक्रीज कर दो. Yes, and they will provide adaptation. Absolutely right. Okay. Yes. So these are the benefits of cross pollination. Now we need to discuss the pollen and stigma interaction. Okay. So let's move on to the next topic. Almost pollination is done. All right. Now, whenever pollen reaches to the stigma, this is the stigma, this is the style and ovary, we all know. Whenever the pollen reaches to the stigma, here I have drawn 3 pollens, they have reached to the stigma. Interaction between these two will initiate the process of formation of pollen tube. Alright, and these interaction will recognize whether this pollen is belonging to the same species or not. Right? Agar same species ka nahi hai, then after this recognition, it will not allow to grow the pollen tube. Right? Yes. So after this interaction, pollen tube grows and process of fertilization is initiated. Now it is seen that इसके ऊपर जो शुगरी सोल्यूशन होता है, स्टिग्मा आपने देखे होंगे स्टिकी होते हैं बिकॉज दे आर हैविंग अ शुगरी सोल्यूशन. अगर आप आर्टिफिशियली भी इस शुगरी सोल्यूशन को स्प्रे कर रहे हो, लेट्स से 10% ऑफ द शुगर सोल्यूशन आप स्प्रे कर रहे हो फ्लावर्स के ऊपर, देन इट इज सीन दैट देयर इज इंक्रीज, एब्सोल्युटली that is recognition, that is increase or what we can say fast growth of pollen tube is there. Okay, so these techniques are used in horticulture. Okay, whenever you are dealing with plants and flowers. All right, so pollen pistil interaction is a safety measure. All right, just to ensure whether the pollen is belonging to the same species or not. All right, yes. अच्छा, अब स्टिग्मा के ऊपर पोलन पहुंच चुका है. नेक्स्ट ये पोलन ट्यूब ग्रो करेगा एंड इट विल रीच टू द ओवम. नेक्स्ट प्रोसेस, नेक्स्ट हेडिंग विल बी फर्टिलाइजेशन. सो नाउ लेट अस डिस्कस द फर्टिलाइजेशन प्रोसेस. Fertilization process. Definitely, fertilization is defined as the fusion of male and female gamete. Male gametes are with the pollen. Remember the generative cell? हमने कल की क्लास में पढ़ा था. That generative cell will travel in pollen tube and reaches to the egg cell. And in the meantime, while traveling, it will undergo one division and will form 2 male gametes. Okay, these 2 male gametes will have their own fate that we will discuss. Let's start the process of fertilization. Okay, now अभी मैंने आपको बताया कि फर्टिलाइजेशन यहां पर पोलन ट्यूब की हेल्प से हो रहा है. ऑलराइट, पोलन ट्यूब जो है वो मेल गैमेट्स को कैरी कर रहा है ओव्यूल के पास तक. This process of fertilization by forming pollen tube is known as siphonogamy, and this is the characteristic feature of Angiosperm, Siphonogamy. Okay, fertilization with the help of formation of pollen tube. Characteristic feature of angiosperm. All right. अच्छा सर पोलन ट्यूब तो बन गई, पर पोलन ट्यूब को कौन बताएगा कि किधर उसका ग्रोथ होना है, उसका मूवमेंट किधर होना है. ऑलराइट, सो उसके लिए एक स्पेसिफिक केमिकल कॉम्प्लेक्स होता है which guides the growth of pollen tube in a particular direction. Okay, that specific chemical complex is made up of calcium, boron, inositol chemicals. Calcium, boron, inositol complex. Okay, this complex, the concentration of this complex is sensed by the pollen tube, and the growth of pollen tube occurs in the direction where more concentration is present of this complex, and this complex is generally released by the ovule. That's why ovule तक जो है pollen tube का growth होता है. You need to remember the name of this complex. All right. Now, because ये chemical complex है और इनके influence में pollen tube की growth हो रही है. All right. That's why this type of movement is known as chemotropic movement. So growth of pollen tube is under the influence of chemical known as chemotropic movement. Right? Yes, filiform apparatus and your synergics, they help in the direction. Okay? In fact, जब एंट्री होगी पोलन ट्यूब की एम्ब्रियो सैक के अंदर, तो एक सिनर्जिट के थ्रू ही होने वाली है. उसमें कंप्लीट सिनर्जिट जो है वो डीजनरेट हो जाने वाला है. ओके, यस. So that was the chemotropic movement under the influence of chemical. All right. Now, sir, जब स्टिग्मा से पोलन ट्यूब ऐसे मूव करेगा तो बीच में बहुत सारे सेल्स आएंगे. हो सकता है स्टाइल के जो सेल्स हैं वो सॉलिड हो, समटाइम हॉलो भी हो सकते हैं, या समटाइम स्पेसिफिकली पोलन ट्यूब के लिए उन्होंने कैनाल्स का फॉर्मेशन कर दिया होता है. तो सर ये बैरियर जो है बीच में, हर्डल्स, इनको पोलन ट्यूब कैसे क्रॉस करने वाला है? तो रिमेंबर दैट यहां पे पोलन ग्रेन है एंड पोलन ट्यूब स्टार्ट्स फॉर्मिंग लाइक दिस. इट रीचेस टू द embryo sac like this. रास्ते में it will release pectinase enzyme. Okay, yes, we are coming on to the micropyle and just wait. Pectinase enzyme, and अगर आपको याद हो तो जैसे एनिमल सेल में सीमेंटिंग मटेरियल जनरली हाइलुरोनिक एसिड होता है, जस्ट लाइक दैट प्लांट सेल में सीमेंटिंग मटेरियल क्या होता है? मिडिल लैमेला, दैट इज कैल्शियम पैक्टिन. राइट? So just to loosen up, just to remove that cementing material so that the growing pollen tube can make their way, right? वो रिलीज करता है पैक्टिनेज एंजाइम, जिससे कि सेल लूजन अप हो जाते हैं और पोलन ट्यूब को एक वे मिल जाता है, right, for the growth. So like this it will grow by releasing pectinase enzyme. Let me write it here. Pollen tube releases pectinase enzyme. Okay, pectinase enzyme. Okay, all right. Yes, it will act on calcium pectate, the middle lamella, and loosen up the cell, making its own way. Now, finally, it reaches to the embryo sac. Let's say ये embryo sac है. It is attached with the wall of ओवरी लोक्यूल्स। अब कल हमने देखा था पूरे ओव्यूल का स्ट्रक्चर एंड डिफरेंट टाइप्स ऑफ ओव्यूल्स आल्सो, एनाट्रोपस, एम्फीट्रोपस, लाइक दिस। अगर पोलन ट्यूब की एंट्री माइक्रोपाइलर एंड से होती है, then this type of entry, this type of fusion is known as porogamy. All right, yes, this is known as porogamy. If pollen tube is entering through the micropilar end, that is known as porogamy. It is entering with the help of micropyle pore. Okay, it is known as porogamy. Now another entry points are also there in the embryo sac. For example, it is entering from the just opposite end of micropyle. That end, कल क्या बोला था हमने उसको बोला था chalazal end, right? So this type of entry of pollen tube from chalazal end is known as chalazogamy. What do we call it? Chalazogamy, right? So next type is chalazogamy. Okay, entry from chalazal end. Chalazal end. अगर आपको याद आ रहा है कल जो हमने पढ़ा था, माइक्रोपाइल एंड चलाजल एंड बोथ आर प्रेजेंट अपोजिट टू ईच अदर, तो ये तो हो गए अपोजिट एंड से एंट्री पोलन ट्यूब की। अगर पोलन ट्यूब की एंट्री इन दोनों एंड से नहीं हो रही है, इन बिटवीन इंटेग्यूमेंट से हो रही है, देन वी कॉल इट मीजोगेमी। व्हाट डू वी कॉल इट? वी कॉल इट मीजोगेमी। I hope it is not going out of the focus. Yes, it is visible. Absolutely right. That was known called as mesogamy. Very good. Okay. Yes. Through integuments or any site other than chalaza and micropyle. That is mesogamy. Okay. Now you need to remember the examples also. For example, porogamy is your lily plant. Lily plant के अंदर porogamy होता है. Then chalazogamy is found in juglans. Okay, juglans. And the mesogamy is found in cucurbita. All right, cucurbita. What happened, Purva? Why you have written beta? No. Have I asked any question? Yes, cucurbita is the example of mesogamy. All right. So that was about the different types of gamy. 3 different types. Okay. All right. Now moving on to the next. That is fertilization के आगे का process. अभी पोलन ट्यूब जो है ओव्यूल के अंदर पहुंच चुकी है, नियर दी एग सेल। मैंने बताया इधर किसी भी एंड से उसकी एंट्री होगी, राइट? इट विल मूव टुवर्ड्स द एग बाय डीजनरेटिंग वन ऑफ द सिनर्जिड। ओके, ऑल राइट पूर्वा। यस, देयर इज अ लैग एक्चुअली, सो दैट्स व्हाई आप लोग जब आंसर लिखते हो मैं बहुत आगे चला जाता हूं, और मैं जब क्वेश्चन पूछता हूं तो आप लोगों को आंसर करने में भी बहुत देर लगती है, और इस बात का फायदा कुछ बच्चे उठाते हैं, राइट? जो आंसर नहीं करना चाहते हैं, जिनको आंसर नहीं आता है। ओके, there is no need to go to a gym when you are cleaning this board. This is quite hard to clean. It will make muscles automatically just by cleaning this. All right. Now, next step of fertilization. Now the pollen tube has reached to the embryo sac by different ways we have just discussed. While moving towards the embryo sac, generative cell divides and form two male gamete, first male gamete and second male gamete. Now, the first male gamete fuses with the egg cell. Okay, and fuses with egg cell to form embryo. It will lead to the formation of embryo. First it will form zygote and then embryo will be formed, right? So here I have directly written embryo. That is understandable. Next is second male gamete. All right, second male gamete. It will fuse with the polar nuclei, okay, which will form the endosperm later on. Okay, so because two different times or fertilization is taking place twice, this process, this phenomena is known as double fertilization. Okay, what do you call it? You call it double fertilization. And again, double fertilization is the characteristic feature of angiosperms. Angiosperm का characteristic feature होता है, right? Absolutely right. Okay. जो ये फर्स्ट मेल गैमीट एग के साथ फ्यूज हो रहा था, फर्टिलाइजेशन हो रहा था, this fertilization is known as, what do we call it? We call it generative fertilization. Generative fertilization. Why? Because it is leading to the formation of embryo. Okay. Second fertilization of this is known as vegetative fertilization because it is not leading to embryo formation. That's why it is called as vegetative fertilization. Okay. Yes, it is not leading to the formation of any embryo. That's why it is vegetative fertilization. So double fertilization involves these two different type of fertilization. I hope it is clear to everyone. Yes, please remember this. Now, the generative fertilization is a type of syngamy. Syngamy, in the starting we have discussed. Quickly reply in the chat box. What do you understand by syngamy? Let's see आप लोगों को कितना याद रहता है. Quickly reply in the chat box. What is syngamy? I'm waiting for your answers. Okay. I will not start new topic until you will reply. Okay, Jamila has written fusion of male gamete with female gamete. Definitely, syngamy is the fusion of male and female gamete. Simple. Okay. Yes. Harsha has written seed fertilization with confusion. She is thinking whether she is right or not. So yes, it is fusion of male and female gamete. Simple. Right, Prathu? oosphere causing fertilization, right Gaurav? Simple, very simple it is, right Nilima? Okay, so that was about the fertilization. अब fertilization हो गया है, next process will be embryo formation, right? So embryo formation is again थोड़ा सा ट्रिकी है, तो थोड़ा ध्यान से सुनना, विल अंडरस्टैंड दिस इन वेरी शॉर्ट फॉर्म. अच्छा, एम्ब्रियो के डेवलपमेंट के लिए न्यूट्रिशन इज प्रोवाइडेड बाय एंडोस्पर्म. एंडोस्पर्म is diploid. Acha, one more thing, important thing. Here the male gamete, the ploidy is haploid, and the ploidy of egg is also haploid. Fusion leads to the formation of diploid zygote. Diploid zygote is formed, which will later form embryo. Here, male gamete, second male gamete, the ploidy is N, haploid. Coming to polar nuclei, already polar nuclei are N plus N, right? When N and N plus N fuses, they will give rise to a triploid structure, right? Therefore, endosperm formed is triploid in nature, okay? And that's why it is going to form the nutritive tissue for the growth of embryo. एम्ब्रियो के ग्रोथ के लिए एंडोस्पर्म जो है वो बहुत जरूरी है. ऑलराइट सर, अगर एनिमल्स की बात करें तो एनिमल में अगर ऐसे ट्रिप्लॉइड बनता है तो वो एनिमल सरवाइव नहीं कर पाता, या फिर उसमें क्रोमोसोमल अबनॉर्मेलिटी हो जाती है. प्लांट्स के अंदर ऐसा क्यों नहीं होता? आपने सुना होगा, हमने वीट की वैरायटी हेक्साप्लॉइड, डोडेकाप्लॉइड तक डेवलप कर दी है. इनफैक्ट सीड्स को सो करने से पहले हम माइटोटिक पॉइजन कॉल्चिसीन प्रोवाइड करते हैं. माइटोटिक पॉइजन मतलब इक्वल डिस्ट्रीब्यूशन नहीं होने देता है वो, राइट, ड्यूरिंग माइटोसिस. उससे क्या होगा, एक सेल के पास बहुत ज्यादा क्रोमोसोम सेट्स हो जाएंगे. इट इज बेनिफिशियल फॉर प्लांट, ऑलराइट, बेनिफिशियल फॉर प्लांट. और एनिमल्स में अगर ऐसा करें तो उनमें डिफरेंट डिजीजेस हो जाते हैं, वो सरवाइव नहीं कर पाते. तो हाउ इट इज डिफरेंट एक्चुअली, क्यों ऐसा क्यों? और ये भी ह्यूमन ने ही, ग्रीडी ह्यूमन ने ही डिस्कवर किया है कि प्लांट्स में अगर आप जितने क्रोमोसोम सेट्स बढ़ाओगे, डिप्लॉइड, ट्रिप्लॉइड, हेक्साप्लॉइड, उतना ज्यादा अच्छी वैरायटी होती जाएगी उस प्लांट की, उतनी अच्छी ईल्ड प्रोडक्शन वो प्रोड्यूस करेगा. यस, तो ऐसा क्यों भाई? प्लांट में तो बढ़ता जा रहा है प्लॉइडी, तब भी कोई प्रॉब्लम नहीं है. एनिमल में बढ़ाओ तो एनिमल इज नॉट एबल टू सर्वाइव. एनी आइडिया, एनी आंसर? यस, let me tell you. See, what happened in plant, जनरली जो ह्यूमन है वो किन प्लांट के ऊपर काम करेगा जिससे उसको कोई प्रोड्यूस मिल रही होगी? डेफिनेटली जिससे कोई प्रोड्यूस नहीं मिल रहा है, ह्यूमन बहुत ग्रीडी है, राइट, बहुत सेल्फिश है, वो उसके ऊपर काम भी नहीं करेगा. तो प्रोड्यूस बेटर मिलने के लिए उसके अंदर मोर न्यूट्रिशन होना चाहिए. मोर न्यूट्रिशन which means more protein, different forms of enzymes, they can synthesize different nutrients. अब ज्यादा नंबर ऑफ जीन्स, ज्यादा अमाउंट ऑफ क्रोमोसोम होने से ज्यादा अमाउंट ऑफ प्रोटीन भी सिंथेसाइज होगा. दैट इज व्हाई प्लांट्स में they are able to survive and we are doing this. But in animals it is not permissible and it is not accepted by nature also. Okay, so that is the reason. All right, now moving on to the formation of endosperm that will help in the growth of embryo. endosperm formation, right? I have just told you that it is triploid. So basically it's a nutritive tissue. It will provide nutrition to the growing embryo, right? Now, again, this endosperm is of three different types in different plants. Let us discuss their type. Number one is nuclear endosperm. Nuclear endosperm. Nuclear endosperm. Name itself suggesting that mainly it is formed of different nuclei. So karyokinesis rapidly or repeatedly occurs in the formation of this type of endosperm, right? This is the embryo sac. Here triploid uh, triploid nucleus is there of endosperm. It will continuously form the more nuclei and pushes the newly formed nuclei towards the periphery like this. So complete space is filled with nuclei mainly. All right. Complete space is filled with nuclei. This type of endosperm form is known as nuclear endosperm. Okay. Now, the example of plants in which nuclear endosperm is found is your maize, wheat, rice, sunflower, etc. Right, Jamila? Absolutely right. So these are the example of plants in which nuclear endosperm is found. Okay. Here we will discuss the example of coconut also. All right. What happened in coconut? Coconut is having multicellular as well as free nuclear endosperm. So the peripheral part is known as multicellular endosperm, whereas the central liquid part is free nuclear endosperm. Okay, that's why it is suggested to have coconut water. All right, it is very nutritive in nature. Okay, yes. All right. So that was about the nuclear endosperm. We have discussed the examples also. Next is cellular endosperm. Okay, cellular endosperm. Definitely, here nuclei are formed. In place of nuclei, everywhere cells will be present. Okay? Yes, cells will be present. This type of endosperm formed is known as cellular endosperm. And the example of plants in which cellular endosperm is found is petunia and balsam. Balsam is having a very delicate flower. Okay, rainy season में अभी बहुत मिलेगा balsam का flower, whitish, pinkish. There is a variety of color in the petals. बहुत ही delicate. So इनके अंदर मिलता है cellular endosperm. Coming to the third, that is halobial endosperm. Halobial endosperm. Now, what happened in halobial endosperm? Both nuclei and cells are present scattered in the region. Okay, so it is formed by karyokinesis, and in some cytokinesis is also followed. So it will give rise to some cell, and many nuclei are also present. That is known as halobial endosperm. And halobial endosperm is found in a plant known as Asphodelus. Asphodelus. You need to remember the example actually because it is different and the example is being asked. Okay. So that was about the endosperm formation. Now your endosperm has been formed. Let us discuss the development of embryo. Okay, development of embryo देखते हैं हम. Development of embryo. I have told you that by the fusion of first male gamete and egg, zygote is formed. So your first cell of the next generation, that is the zygote. Okay. Now this zygote will divide into two different cells like this. Two different cells. The one which is present towards the micropyle end, all right, towards the micropyle end is known as basal cell, and the one which is present away from the micropyle end is known as terminal cell. Terminal cell, also known as embryo cell. Okay, basal cell and embryo cell. Okay, also known as terminal cell. Now, this basal cell will going to form suspensor. All right, and the last cell of suspensor is known as the hostorium. All right, like this. Basal को हम divide करते जाते हैं continuously 7 to 10 around, और इसका जो last cell है वो थोड़ा सा बड़ा होता है. This is known as hostorium. All right, and this line of cell is known as suspensor cell. Suspensor cells. Okay. Now, at the base, there present the terminal cell. Now, the terminal cell also divides. Okay. How the division takes place in terminal cell? First of all, one vertical division. Okay, this vertical division followed by a transfer division. Okay, this is followed by a transverse division. You are observing 4 cell, but after one more division, these 4 cell will become 8 cell, and they are arranged in 2 tiers. Okay, tiers, you know, एक के ऊपर एक layers के form में. Yes, absolutely right. Sound is cracking. Check your internet, beta. Must be the problem by internet. Otherwise, बाकी लोगों के लिए sound सही है. All right, not an issue. So they are arranged in 2 tiers. Okay. Yes. The layer which is present near to the suspensor is hypobasal layer. Hypobasal layer. And the layer which is present away from the suspensor is known as epibasal layer. Epibasal layer. Okay, so now 8 cells are formed which are present in 2 tier. Okay, 2 layer. Okay, that's great. All right, now slowly this part of the cell, this part of the structure will divide more. All right, it will undergo mitotic division to form the complete embryo, and finally it will form the structure like this. इसी में मॉडिफिकेशन करते हैं क्या? चलो इसी में मॉडिफिकेशन कर देते हैं. अच्छा, ये जो लास्ट सेल है सस्पेंसर का, which is connected with the embryonic mass, is known as hypophyseal. Okay, or hypophysis. Hypophysis. All right. Now here the division takes place and structure like this is formed. Here one cell is there. Here one layer of cell is present. Okay. There is another layer present. Okay, there is another layer present, and inside that there present one more mass of tissue. I am using another pen. Okay, so this another mass of tissue is present. Now let us label the different layers of this embryo. Okay, so the cell present near hypophysis will going to form the root cap. This will going to form the root cap. Okay, and the next cells which are present here are going to form the root tip. Okay, the outermost layer is known as dermatogen. All right, which is going to form the epidermis, and then we are having the ground meristem. Okay, the ground meristem, this ground meristem, also known as ground meristem को हमने बोला है पेरीब्लेम. And the inner is pleuron. Okay, and pleuron is procambium. It will going to pleuron layer. Okay, so this is how the structure is formed. Absolutely right. Now this part will again start dividing very fast, very rapid. Okay, and it becomes heart shape. And when it becomes heart shape like this, these two parts will grow more fast and it will protrude outside like this. Okay, ये ऐसे grow करने लगेंगे, and this will form two cotyledons. This will form two cotyledons of the embryo, and between cotyledon, plumule will be present, right? So let me draw the structure here. How it will look like when it will grow completely. This is ostomium, suspensor cells are there, and the embryo will grow like this. Okay, and here at the center it will have plumule. Okay, and these two are cotyledons. All right, and this part which is present near hypophysis forms the radicle. All right, so this is how the different parts are formed in the embryo. All right, I hope it is clear to everyone. Yes. Okay, now that was because two cotyledons are there. So it is clearly visible that it is the development of dicot embryo. What happened in monocot embryo? Single cotyledon known as scutellum. All right, single cotyledon is formed known as scutellum. Scutellum. All right. Yes, they are formed. All right. And plumule and radicle, they are also covered by a membrane known as polyorhiza and polyoptile. Okay, shoot apex is covered by polyoptile and the radicle is covered by polyorhiza. Okay, yes, so that is the main difference actually, and that is how the development of embryo takes place. Okay, all right. After that, cotyledon will form, they will grow further and they will form the seed leaves within which the embryo is being enclosed. Okay, yes. So ये पूरा प्रोसेस था एम्ब्रियोजेनेसिस का इन प्लांट्स. अच्छा, वन इंपॉर्टेंट अनाउंसमेंट बच्चों, कल के टेस्ट में सिर्फ ह्यूमन रिप्रोडक्शन आ रहा है फ्रॉम फीमेल रिप्रोडक्टिव सिस्टम टिल एम्ब्रियोजेनेसिस तक. ओके, तो कल के टेस्ट में सिर्फ इतना ही आ रहा है. अच्छा, इसके बाद जो है सीड डेवलपमेंट, फ्रूट डेवलपमेंट, ये मॉर्फोलॉजी में भी देखना ही हमको, तो ये टॉपिक हम यहीं पे खत्म कर रहे हैं. ऑलराइट, यस, एंड फ्रॉम मंडे विल स्टार्ट न्यू टॉपिक. बिफोर दैट यू नीड टू अपीयर इन द टेस्ट. यू आर गेटिंग good collection of MCQs. Solve them honestly. Send me your score. All right. So that's all for today, guys. We are ending our class here. See you in the next class. All the best for your test. Thank you. Bye-bye.